Welcome to the Biochemistry Clinic. Today I want us to look at the TCA cycle, which actually occurs in the matrix of the mitochondria. Now, I want us to do something a little bit different today. I want us to begin with the end in mind. So at the end of the TCA cycle, what exactly do we stand to gain? So we're going to generate one GTP molecule, which is actually equivalent to one ATP molecule. We're also going to generate one FADH2 molecule, which is equivalent to 1.5 ATP. And we're going to produce 3 NADH molecules, each of which is equivalent to 2.5 ATP molecules. And when this is multiplied by 3, you're going to have 7.5 ATP molecules. This simply means that for one round of TCA, when you invest one acetyl-CoA molecule in the TCA cycle, you are going to produce 1 plus 1.5 plus 7.5 ATP molecules, which should give you a total of 10 ATP molecules, which is quite a lot of energy that we stand to gain from the TCA cycle, of course, with carbon dioxide that is actually produced. So here's a quick practice question for you to just keep yourselves busy. Calculate the energy produced in the TCA cycle in terms of ATP equivalents from one molecule of glucose. So we've just calculated the amount of energy that is produced from one acetyl-CoA molecule. What of one molecule of glucose? So this takes us back to glycolysis. The oxidation of one glucose molecule produces 2 pyruvate molecules, which will produce one acetyl-CoA molecule each in the mitochondria. So meaning that one molecule of glucose is going to give you 2 acetyl-CoA molecules in the mitochondria. And we know that one acetyl-CoA molecule produces 10 ATP molecules in the TCA. So therefore, for one glucose molecule is simply 2 times 10. But out of curiosity, I would want you to try something, and I want you to actually post your answers in the comment section. So I want you to calculate the total ATP yield from glycolysis to the TCA cycle. So I'll give you a clue quickly. In glycolysis, we produce 2 NADH molecules, 2 ATP molecules, and 2 pyruvate molecules. And the other clue that I need to give you is that when pyruvate enters the mitochondria for the decarboxylation, which produces acetyl-CoA, each conversion of pyruvate to acetyl-CoA produces 1 NADH molecule. So please calculate the total ATP yield from glycolysis up to the TCA cycle from one molecule of glucose. When you find the answer, please drop it in the comment section. Now, let's quickly go into the steps. Now, TCA occurs through a series of steps, and these are 8 different steps. So I'll quickly just point out what you should always focus on. Now, in the first step, what you should know is that acetyl-CoA, which is actually coming from decarboxylation of pyruvate, is combining with oxaloacetate to produce citrate in step 1. What are you supposed to know about step 1? Step 1 is catalyzed by citrate synthase, and citrate itself can actually inhibit this enzyme. So this is also catering for the regulation aspect. So please take note that citrate itself can actually inhibit. So when you have too much citrate, this enzyme citrate synthase will actually be inhibited so that this reaction does not proceed in the forward direction. In the second step, we just have citrate being converted to its isomer, isocitrate, and the enzyme is aconitase. Why aconitase? Because there's an intermediate structure which we call the cis-aconitate between citrate and isocitrate. That's why the name of the enzyme, for those of you that struggle with the names of the enzymes, that's why the enzyme is called aconitase, because of the intermediate, the cis-aconitate intermediate. Step 3, which is very important, I think the most regulated step of this pathway. So you have to pay particular attention to it because it determines how you can control this TCA cycle. So in step 3, you have isocitrate being converted to alpha-ketoglutarate. And the enzyme is isocitrate dehydrogenase. What else are you supposed to know about step 3? Remember those things that we are producing, the summary that I gave you. This is why you produce your first NADH. And also carbon dioxide is produced. This is a decarboxylation reaction. So a 6-carbon molecule isocitrate is converted to alpha-ketoglutarate, which is a 5-carbon molecule. Why? Because we are getting rid of carbon in form of carbon dioxide. Then we move to step 4, which is similar to step 3 actually, because here we also have decarboxylation, which gives us carbon dioxide, and we also have NADH produced. And the enzyme here is alpha-ketoglutarate dehydrogenase. So you see already in step 2, in step 3 and 4, we already have some of the products that we started with. So just take note. Step 3 gives us the first NADH. Step 4 gives us the second NADH. So we've produced 2 NADH molecules just up to this point. Then in step 5, we have succinyl-CoA being converted to succinate. And here again, we appreciate the production of GTP, which is one of the things that we talked about in our summary. And the enzyme here is succinic thiokinase. Then we move to step 6. Step 6 of the TCA cycle involves the conversion of succinate to fumarate. What are you supposed to know about step 6? FADH2 is produced. Remember, one of the products that we talked about in the beginning is produced in step 6. And the enzyme here is succinate dehydrogenase. And if you've noticed, while we are producing these electron carriers, NADH and FADH2, the enzymes, those of you that struggle with the names, just focus on the names. This is succinate dehydrogenase. Remember, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase. So we are producing those reducing agents or the electron carriers, NADH and FADH2. There's something that you should pay particular attention to the names of these particular enzymes. Then in step 7, fumarate is simply being converted to malate with the help of the enzyme fumarase. And lastly, in step 8, malate is converted to oxaloacetate, which is one of the starting molecules. And of course, what you should know here is that we are actually producing our final NADH molecule. So now you can even count these molecules that are actually in green will help you understand our initial summary here that we started with these products of the TCA cycle. Now, just something quick about the regulation of the TCA cycle. So the regulatory points are usually step 3 and partly step 4. Of course, we've talked about step 1 also, where a lot of citrate can actually inhibit. So here, when you have a lot of NADH and a lot of ATP, so I want you to know, and this is very clear. Why should a lot of ATP inhibit isocitrate dehydrogenase? Because the body does not want to continue producing ATP even when you have too much. So remember, what inhibits the TCA cycle? An abundance of ATP, an abundance of citrate, and also NADH. These will inhibit the TCA cycle. The reason is very simple. The body does not want to overproduce ATP and to waste. And of course, we know that this can actually be activated by ADP, the presence of ADP. So if you have a lot of ADP present, it means that you can have actually the TCA cycle being activated. So if you have any questions, please drop them in the comment section so that we can actually address them in the next video. Thank you very much.